Professor Rezsöhazy. Today we have the honor to interview you, and I will briefly introduce you and your career path. Please feel free to correct me if I get anything wrong. So you studied biology here in Louvain-la-Neuve. You also completed a PhD in molecular genetics, focusing on transposable elements in bacteria. And afterwards, you carried out a postdoctoral fellowship in Amsterdam, in the Netherlands, Cancer Institute, still in transposable elements, but this time in animals. At the end of this postdoc, you returned to UC Louvain, where you joined a research group working on molecular embryology, and before funding your own team in this field, in Louvain-la-Neuve, in parallel, you also took on the responsibility of teaching to students. And so to start this interview, could you give us a short overview of your research topic? So why is it important, why you started it, and also for those who maybe are not specialists of that topic, could you explain why is it so interesting? So yeah.
Okay, thank you, Pierre. So about my current research activities, so we study in my lab genes, which are extremely important to build up an embryo, an animal embryo, and these are called architect genes, because if you mutate those genes, you will have problems during the embryonic development. So those genes in particular code for proteins, like lots of genes, and those proteins control other genes. So this is a bit weird. These are genes coding for proteins that control genes. And this is why they are architect, because they will tell, those proteins will tell in the cells, yeah, you are contributing to develop a head or a limb or a vertebra, and all together those genes will shape the body of the animals. So what in particular we study in the lab, it's the mode of action of those genes. And somehow this is very basic, very fundamental, because this allows us to understand how the embryonic development takes place. Now there is an interest in biomedical sciences or in the clinic, because those genes can be mutated in the human population, and this leads to malformations, to developmental problems, but also to cancers. Because as you know, cancers can be viewed somehow as pathologies of developing and multiplying cells. So it is very basic, and it's very exciting to understand how an embryo, an animal embryo, is built up. But there are interests for the clinic. Yeah.
Okay, so it's always interesting to have the point of view of the principal investigator. And I would say, what do you enjoy the most about being a principal investigator?
Well, of course, the first credo we have as PI in a lab, it's to make research. And from my point of view, making research, it's always pushing the borders of knowledge. In the lab, on a daily basis, any student, master students or PhD students or postdocs, on a daily basis, they discover new things. Not great things, not big things, not things that will make a Nobel Prize, but on a daily basis, the students in the lab, so they see some stuff they are the first to see in the world, in the history. So pushing the frontiers of knowledge, increasing the knowledge about specific topics like in biology, embryology, etc. This is the real excitation. So this is really the stuff I love much. Now, in parallel, of course, research activities are connected to teaching, to transmit the knowledge. So you build up the knowledge and then you transmit the knowledge to new generations, to students. And this is also exciting, because when you have students, bachelor students, master students, and you tell them your story or the history of science, you see in their eyes, that, wow, excitement as well. So making research, teaching, these are connected activities. And all this is around building knowledge, crossing the borders, and transmitting this to the new generation.
Perfectly summarized. I had a few questions for you, more specific to particular devices. So here we do synthetic biology in the iGEM competition, and so far new sequencing methods, CRISPR-Cas9, AlphaFold, AI, really helped us in these projects. And how these innovation change the way you work, or maybe your colleagues, because if you don't really use AI or those tools, but do you think it does reshape the very nature of the researcher role in some ways?
Okay, all these technological developments have really changed science, the way we build up models, etc. But the question you raised here is more profound, because you said that if those technologies have reshaped the nature of science. I don't think so. Not the nature of science, but the way we generate knowledge, yes. And the way we generate knowledge, meaning the way we build up models, because generating knowledge always relies on building up models. Talking about AlphaFold, it's a new way to build up models. CRISPR, the recombination tool, it's also a way to build up a new model. For example, new cell lines which are recombinant for something, mouse lines which are recombinant for those or those genes. And in that way, all these techniques change the way we can make models and then build knowledge. And very practically, the answer is yes, in my case, because we already use CRISPR and before that, the RNA interference. AI, not yet, because I'm not at all an expert, but I'm sure that the PhD students in my lab use AI more than me, and they are better than me in playing with all those databases and tools, etc. If you look in my office now, there is a big poster with 3 big networks of interactors of my favorite proteins. It has been, those 3 posters have been generated by the PhD students. So, okay, the way to build up this network is a model. The way they have built up those networks, it's thanks to the new tools. Okay, there are tools I don't use because what we study do not rely or do not require this. For example, AlphaFold. In particular, the transcription factors are proteins which are disordered, so AlphaFold is useless in those cases. But yes, definitely, all these techniques, all these new methods contribute to change the way we make science. But my feeling is that they do not change the nature of science.
So it's opening doors, it's maybe a new tool in the box, but it's never going to change, at least for now, the researcher role and nature of the scientific research itself?
No, I don't think so, yeah.
And those technologies definitely raise ethical concerns.
Yeah.
And at least I've seen it with you, because you give philosophy and ethics in master degree for a scientist. And so they can be used, both those technology, to save life and potentially to cause greater harm. And so double usage is often...
Yeah.
So what is your perspective on this? Are you worried that there is maybe some misuse of those technologies?
Okay. For what concerns biology and molecular biology, I don't feel so much concerns of dual use or misuse. I do not see really how those techniques could be perverted towards a bad use. If you look at CRISPR, well, there is nothing new if you compare to any kind of recombinant DNA techniques. So no, and no, I'm rather optimistic about that. All these techniques that allow to appraise big data sets, to AI, etc. In the field of biology, I'm not so afraid. There are other fields of science and technologies for which I'm more afraid. For example, embarking neuromorphic AI in drones to make war, this is something else. And we are close to that, to be honest. So now to answer more broadly, any kind of new technology can supposedly raise ethical concerns, any kind. And this is why we have those kind of courses in ethics for scientists. There are always questionings that deserve to be discussed and appraised. But for what concerns precisely the new methods we apply in biology, for me there is no big risk and problems, no. I don't think so. I don't think so. And this is something weird, but in general, the technologies connected to life sciences, they are very, very well, how to say that? The men in the street and the society and the politicians are very aware and are very cautious about, because dealing and touching and manipulating life concerns immediately human beings, because we are life, we are living organisms. And of course, using CRISPR in medicine could raise some concerns. But CRISPR has already been used in medicine to cure very, very severe and harmful illnesses. And this is positive. And there are ethical groups, bioethics groups in most countries, I suppose. At least in Belgium we have a committee for bioethics, and they do quite a good job. So there is a real care and a real attention to these possible misuses, and this makes me optimistic about that. And this is probably particular to anything like modifying life, because anything that could modify any kind of organism could also modify a human being. Okay, now, of course, the zero risk does not exist, and if you remember these CRISPR babies that have been generated in China, okay, this is the bad example.
Could you maybe explain what is that phenomenon or case? Maybe people don't know it yet.
Yeah, okay, the point is that with the CRISPR technology, so CRISPR are sort of molecular scissors that allow to target genes and to modify specifically and very precisely genes. So you can modify potentially the heredity of any kind of organism, from bacteria to animals and plants. And it is already a very old concern that when in the 80s, 90s, recombinant DNA techniques started to be applied to animals, people said, okay, now it is possible to also modify the heredity of human beings. And ethically, it has been something that was really clear. We can use cell therapy, gene therapy to cure someone, but without influencing his or her potential heredity. So modifying the genome of a human being to come has been something which has been largely forbidden. And the case here in China, a couple of years ago, someone has decided to use the CRISPR technique to modify the genome of a very, very early embryo. And then this embryo was, re-implanted in a pregnant lady, and this embryo became a baby, and now those are girls who have received targeted mutations, and this will influence their life and all their progenies for generations. So yeah, it has been done at least once. At least we know it has been done once. We know that some labs, weird labs around the world, have the same kind of projects, but it has been banned by everyone, by all public governments and all ethical societies and ethical committees. I think that the potential hazards, risks, and dangers are very, very carefully checked by political bodies and bioethic committees, etc. Yeah.
At least in Belgium, we are in a nice place, that's for sure.
Yeah. I know some people who work for the National Bioethic Committee, and they do very good job. I mean,
fantastic.
Yeah, very good. Yeah.
Looking back, what has been the most memorable day of your scientific career?
Difficult to say, but maybe one of the most important was not related to science, but more related to the career. It was the day I was hired with a permanent position. This is not trivial, because you know that the career paths in science are hectic somehow, because you know, from my first graduation with my master in biology, and between the end of my master classes and the day I got really hired for a permanent position, I've been, 16 years on short-term contracts. And if you want to make projects in your life, in particular I'm married and I wanted to have a family, etc. If you want to buy a house, you have to borrow money, you have to deal with a bank, etc. If you keep all the time on a rather unsafe position because you just have a 6 month, 12 months, 2-year fellowship, it's harsh, it can be harsh. So one of the best happening in my life was that now what you have dreamed about for years, that meaning that you could make science and to teach for the rest of your life, when I got this good news, it was really, really cool. Now, talking about science, more typically the discoveries, the modest discoveries we made. I have a few memories. One during my PhD thesis, when at that time we make sequence alignments, to compare the sequence, but we did it by eye, not with computers. And at some point I said, oh, there is something behind this. And we made a very good paper, it was a good paper. It was really the heart of my PhD thesis. When I got this, I said, okay, now my thesis is on the safe side, and this is cool. I had the same kind of excitement or happiness in terms of accomplishment with my Hox genes twice, because we made bets about what could be the phenotype of modifying this gene. Because when we decide to make recombinant organisms, this is not something you decide just like this because, okay, no, it's difficult, it is costly, it requires time, and when we had what we believe to be robust enough hypothesis, we go towards the in vivo. And twice we made the good bet, and the last days here, a couple of years ago, when we identified mutations in patients together with a team in Marseille, mutations that were associated with heart malformation. Mm-hmm. We studied all the molecular biology of the mutation, and at some point we decided, okay, let's try to see if the hypothesis is good, meaning that it's not simply a correlation, but a causal relationship between the mutation and the heart malformation. With CRISPR, we decided to make a single point mutation in the mouse, and we reproduced perfectly the malformation of the heart observed initially in the clinic. I said, okay, wow.
That must be a feeling.
Yeah. We got it, and this is nice. And this is modest, I mean, there is nothing like a big discovery that will bring us to the Nobel Prize or whatever, but you said, okay, now, you end up the loop, you had a hypothesis, based on observations made in the clinic, you make all the cell and molecular biology, you generate an animal model, and you end up with the same malformation and with almost the same penetrance, etc. And now we can study the model because we validated this model as a good model for the human pathologies, and this is a good memory. So you have, as a scientist, good memories and good days because of your career, because those careers are not easy, and also good days because you said, okay, now I had a good feeling, and you validate your hypothesis. It's always cool.
It's always a very hard path to take in terms of research, and then when it finalizes, you can really feel the joy by like all those hard work being done. Well, thank you for coming to this interview, and it was nice having you here. Great answers all the way through, to which I agree most of the time. Sometimes I'm a bit more pessimist, but I would say that it was quite phenomenal. Thank you.
And thank you, Pierre. Thank you.